much for having me to speak today. I feel a bit intimidated being the first one up, but yeah, it's really great to be here, and I'm really excited to see the other talks for the rest of the day. So let me move over here. My name is Grace Power. I'm a research fellow at the IU in Bristol, and today I'm going to talk to you about a project that we've been working on using genetic epidemiological methods to further unpick the relationship between body size, used as a proxy often for BMI, and breast cancer from a life course perspective. So just quickly, I'm going to summarize the presentation. So I'll be going through what life course epidemiology is, kind of give you a quick overview of Mendelian randomization and why it's really useful within this context. Look into the kind of application of MR within the life course setting and the assumptions or the additional assumptions we need to make to be able to utilize this method. Kind of give you an overview of the current evidence within the field. So the current evidence being that we see a predictive effect of increased body size in childhood on breast cancer risk. And then explore the expansion of this into deriving additional methods to better or to look at the effect of BMI at additional life stages, utilizing lots of different cohorts and bringing more data together to be able to do that. So diseases diagnosed in adulthood often have antecedents early in the life course, and gaining a better understanding of how exposures affect outcomes at different points in the life course is really key if we want to improve disease prevention and treatment strategies. And so whilst I think epidemiology is a really compelling framework to kind of work within from an epidemiological perspective, it's really difficult, not only for issues around data, so things that we deal with in epidemiology, like censoring, follow-up when we're working with longitudinal sets, but also insidious types of confounding, like time-varying confounding and intermediate confounding, which are very difficult to identify and then very difficult to deal with. So that's where Mendelian randomization is potentially a really useful method to use within this context, because MR is a method within the kind of causal delivery space that seeks to mitigate unmeasured confounding. And MR, just quickly, for those who don't know, is an instrumental variable analysis. It uses genetic variants as instrumental variables to look at the causal effect of the modifiable exposure on an outcome. And we use genetic variants because they're randomly allocated to us at conception from our parents, and they kind of mimic a randomized control trial as a result. But the genetic variants we use need to be strongly associated with the exposure of interest, not share a common cause with the outcome under investigation, and not affect the outcome other than via the exposure that we're interested in. And so when we do have genetic variants that we use as these instruments, we're able to reduce susceptibility to unmeasured confounding factors, including undiagnosed disease, confounded by undiagnosed disease, which we often refer to as reverse causation. But we do need to make an additional assumption if we want to apply MR within a life course setting, and that is that genetic variants have different effects on the exposure at different periods in the life course. Those genetic variants that we're using kind of need to follow this assumption. And I've just shown here, this is what we call a polygenic risk score plot. So here we've used genome-wide significance SNPs from an adult bus, and we've looked at how well these predict BMI at different points in the life course. And as you can see, they predict adulthood BMI, so that's age in years at the bottom, really well, but really poorly predict BMI in childhood. So that's quite compelling evidence for us that there are going to be genetic variants that do kind of change across the life course. And so by leveraging these, we're able to derive time-specific effects on outcomes. And just to kind of briefly say, I'm a part of the Life Course US Consortium, where we're kind of bringing together data from lots of different cohorts around the world to have better instruments to kind of do this and kind of expand beyond anthropometric exposures. But for the moment, we are kind of focusing on anthropometric kind of exposures. And this slide is just to say there's collaborators of ours in Bergen who are really working hard on better understanding the genetic architecture and what's really going on with these genetic variants at different points in the life course. So a couple of years ago, I was really interested in and did a systematic review to identify Mendelian randomization methods that are utilised within a life course research setting. And in doing this, I identified several trajectory-based methods, which I'm sort of starting to use more now. With I've got some small grant funding to be able to kind of explore these sorts of methods. But there was also a kind of like a core set of methods that we used to better understand sensitive periods. And by that, that just kind of follows the kind of life course hypothesis that simply assumes that we are more or less affected by exposures at different points in the life course on an outcome. So we sort of focus on that sensitive period as being the period that's the most important, perhaps. And within that, at the time, there were a lot more methods since that review, but there were sort of two core methods. There was the structural mean modelling method, and there was this method, which we use a lot at the IU and is pioneered by a colleague of mine, Eleanor Sanderson. I'm going to just refer to this as the inverse variance weighted multivariable MR method, but that's a bit of a mouthful, so I'll just be referring to this as either IWMVMR or NVMR. But so I'm using actually this just to kind of set the context of an applied example, but I'm focusing on cardiovascular disease. I think this sort of showcases how important this is from a public health messaging perspective. So what we do within this methodology framework is genetic variants strongly associated with, for example, childhood body size, genetic variants strongly associated with adult body size, and genetic variants that are strongly associated with both. So these genetic variants are kind of the correlation. And yeah, so IDW is able to estimate the effect of several strongly correlated exposures on an outcome, accounting for the other exposures in the outcome. So if we were interested, and we were with the cardiovascular disease risk example, the effect of increased childhood body size and cardiovascular disease risk, and in a univariable setting, so without accounting for adult body size, we saw that increased childhood body size, increased risk of cardiovascular diseases, the full suite of them. Now, when we accounted for adult body size, that effect completely attenuated. So what we can conclude from that is that it's adult body size that mediates that effect. And the public health messaging there is that we can then intervene around childhood and reduce, if we reduce weight around childhood, then cardiovascular disease risk might be mitigated. Now, the public health message changes slightly when we come on to breast cancer, but I just included this slide to sort of say that assumptions need to be expanded on to include additional exposures in the model, but I'm not going to go into the technical details of life course and life assumptions. And so colleagues, Tom Richardson, Alan Sanderson, and George use this method, and they utilize the UK Biobank data. UK Biobank data being kind of collected around 56, say that's the average age of participants. So their BMI data, but then also the data from their recall of their body size around age 10. And these were, so individuals were asked, how large were you around age 10? Was it thinner, about average, or plumper? And amazingly, when Tom G was these, he validated the instruments across several different cohorts, and they validate really well. So they really predict BMI at younger years very well. And so we've been using these measures to, like, ever since this paper was produced. He sort of in the original paper looked at several outcomes, coronary heart disease being one, type 2 diabetes, breast cancer, and prostate cancer. And so that brings me on to the breast cancer question. So what he saw was that increased early life body size was protective against breast cancer risk in a univariable setting. And he found the same with increased adult body size in a univariable setting. In a multivariable setting, when he accounted for adult body size when he was interested in childhood effects and accounted for child body size when he was interested in the adult effects, he saw that protective effect from childhood body size remained, if not got stronger, but the effect of adult body size completely attenuated. And I just want to say this is what I'm referring to as later life adult body size, because I expand these age periods to look at the sort of early reproductive years in women. And I just also want to say that these findings have sort of been replicated across really good conventional epidemiological studies as well. So we really believe in this increased childhood body size effect being protective. But we don't know what's going on after childhood body size. So we're sort of stuck at this maybe prepubertal age period and sort of late life age period or postmenopausal period. And so I was interested in further exploring post-menarche to around, say, 40. So working with analysts from several different cohort studies, including ICEP, Hunt, MOBA, Generation R, and Generation Scotland, we GWASed BMI in nulliparous women, and I chose to do this in nulliparous women, or we chose to do this in nulliparous women, to get away from some of the confounding effects of having a child on both breast cancer and body size. And so, yeah, we GWAS these in post-menarche to up to 40 women. And we split these up into subgroups. So menarche 20, 20 to 30, and 30 to 40. Obviously, the biggest group was the menarche to 40 group, but so I think that provides the most reliable evidence. So this is going to get a little bit technical, and I don't know how relevant it is to kind of go into the details of the slide actually, other than just to say we identified SNPs from doing this exercise that had different effects at different points on the life course on BMI, and that's just showing these different patterns of effects, and I can talk to you about those later. And then this is a slide also just to say, obviously, we're looking at BMI or body size as a proxy for BMI, so it's the same trait, so they're going to be highly correlated. But within the sort of, when I looked at the genetic correlations between the different body size GWAS, I don't know if you can really see that here, but those that are kind of furthest apart in age have the lowest genetic correlation score, and those that are closest together in age have the highest genetic correlation score, which is quite comforting and kind of led me to have a little bit more faith in the fact that there are different genetic variants kind of playing a part at different points in the life within the same trait. So these are just the models I kind of had previously, but I guess I'm just putting these here to reiterate. We went on to run downstream analyses using Mendelian randomization with these GWASs and the instruments derived through these GWASs. And when we use them within a universal setting, we're getting the total effect. So that's just the effect of increased BMI at that age period on breast cancer, not accounting for any other time, the effects of BMI at any other time point. And then the direct effects are we called the effects that we derive through the multivariable MR direct effects. And so we're able to just get the effect of BMI between menarche and 40 or menarche and 20, 20 to 30, accounting for childhood body size and accounting for later life adult body size. So these are the results. So the top set of results are the full set BMI in nulliparous women between menarche and 40. So an increase of BMI was protective of breast cancer and several different subtypes in a univariable setting. But when we accounted for adult body size, and when we accounted for late life adult body size, those effects kind of remain the same. So adulthood body size is still not really having an effect on breast cancer. But when we account for childhood body size, we see these effects attenuate. And in the kind of broader age group, they don't completely attenuate. So there's something going on in the early adulthood period as well, in indicating a protective effects on breast cancer. Now, the subgroup analysis looks weird, and we've included it and kept it in the manuscript for transparency. but we just don't have enough power we're incorporating a childhood body size GWAS with 250,000 individuals, and the subgroups have, 10 to 30,000 individuals, which is just not comparable, and it's probably doing funny things with the MR model, which we're investigating. But I think what's really interesting is the broader results, the broader age group results, sorry. So what I did also want to say is here the public health messaging is slightly different. So what we really want to do, and what I've got some more funding to do, is kind of unpick the pathways and look at what's going on. What is it that is causing those protective effects on breast cancer? We're not going to kind of have a public health message that suggests you should increase your body size at certain ages and then reduce it to be protective of, it's more, what different hormones are on the pathway and what are the mediating effects really happening. So that will be really interesting to look further. And that's why I mentioned the Life Course GWAS Consortium, because we're bringing data sets together to hopefully have enough data to be able to look into that in more detail. So just this is just a kind of strengths and limitations slide. One of the biggest strengths is kind of looking at an age interval that hasn't been looked at before within this kind of context. Bringing together cohorts, and the resource will be available to look at more diseases or women's health outcomes. But like I said, we had limitations, which was smaller sample size in the UK Biobank, which resulted in lower F statistics and conditional F statistics in the MVMR. And we were restricted to European ancestry because of the cohort groups that we used, which is limited generalizability, and something, unfortunately, I am saying a lot, but we're hoping to change, and we're working to build data sets with more diverse data. So the kind of take home really is that protection likely reflects the combined influence of adiposity in childhood and in the early reproductive years. And early life is a really critical window for breast cancer prevention. And I hope I'm within time. And these are all the people that were a part of the project that I want to thank. Obviously, the analysts from each of the different cohorts contributed a huge amount of their time. So very grateful for that. Thank you.
Well, that's fantastic. Yeah, really well explained. It's quite complex stuff, but you explain MR and then the kind of the findings really well. One question, yeah. Oh, well, you, and then we'll take two questions. Okay, first David.
Thank you. So the correlation between increasing BMI and protective effect, was that sort of linear throughout the whole range? I.e., so at the lowest BMI, you're at greatest risk, and it just sort of progresses all the way through. And if so, how do you sort of square that with public health messaging? Yeah,
so we assume a linear model, but what is more difficult actually in relation to the childhood body size situation is that we have this, like I said, this prepubertal body size measure, which is like this 3 category. We assume a linear increase, but that's a categorical thinner and about average and plumper, and that about average group is the biggest group. So yeah, it's a little bit complicated. Yeah, then kind of just the sort of like linear trajectory of BMI in the context of this categorical childhood body size situation.
Thank you.
It's not unlike that question. There's so much, as you were saying. You've got to be slim, you've got to be this,
yeah.
Your BMI's down, and now you're beginning to come round, and this is just to do with breast cancers. Can you see this being projected to the likes of the bowel cancers and other major cancers, that maybe being skinny is actually, say slim, should I say, having a lower BMI is actually going to be counterproductive, and it's totally going to toss the health messages out?
Yeah, I came across the same issue when I was looking at fracture risk in my PhD, because an increased body size was shown to be protective against fracture risk in later life, because
more padding.
yeah, well,
Okay.
So in childhood, your bones are developing, and so your bone mineral density is getting much bigger and you're getting stronger. And so when you have that happening in childhood, it's actually really beneficial. But of course, the public health messaging, because of adverse health outcomes in relation to, cardiovascular or like other kind of morbidities and mortality, we can't kind of push the, yeah. Yeah, and the other thing actually to say is we were kind of unpicking a different methodological suite, which was the structural mean modeling MR work. And we sort of started to realize like how communication was like, we were really chopping up body size as if you didn't have an experience of your childhood body size when you were an adult, And I guess my point in saying that is we try really hard not to get too into the methods to make the kind of messaging accessible and actually mean something. But yeah, there have been instances where we've kind of had to take a step back and think like, what is it that we're wanting to communicate here and what's going to be valuable in a clinical setting?
Yeah. Thank you very much.
Thank
Sounds
you.
great. Thank you.